Good morning, everybody. Thank you for joining us. We have Dr. Bengi Baran here today for our KCNI speaker series. Dr. Baran received her PhD in psychology from the University of Massachusetts Amherst, where she studied the effects of aging on sleep-dependent memory consolidation. Then she went on to complete her postdoctoral research training in sleep, circadian and respiratory neurobiology at Harvard Medical School under the mentorship of Dara, Manoach, and Stickgold. Prior to establishing her lab at the University of Iowa's Department of Psychological and Brain Sciences, she served as research faculty at Massachusetts General Hospital. Dr. Baran is interested in understanding the role of sleep in etiology of psychiatric disorders. She also serves as the Director of Neuroscience Research of the Psychosis-risk Intervention, Education and Research PIER program, a research and clinical service program that aims to enhance our understanding of at-risk mental states, identify neuroscience-informed predictors of conversion, and dramatically reduce the duration of untreated psychosis in the community. Today, Dr. Baran will be talking about understanding the role of sleep oscillations in symptoms and cognitive deficits in psychotic disorders. Dr. Baran, the floor is yours.
Thank you so much, Rubaya, for this kind introduction. And thank you, Sean, for this invitation. I've been following you guys' group for a while quite enthusiastically, and it's great for me to be here to share my pre-work and see how we overlap and how we can inform each other, hopefully. So the work that I chose to present today relates to sleep spindles and schizophrenia, which spans both my postdoctoral and independent work. So Rubaya did a great job of explaining my pedigree, but just to show it on the map, this is where I'm from. And I have to acknowledge two great mentors that I had as a postdoctoral researcher, Bob Stickgold and Dara, Manoach, who started this work at the time that I moved to Boston. A couple of years ago, I moved to the University of Iowa to establish my lab. I directed neurodevelopment and psychopathology lab here, NAP lab, because every lab has to have a cool acronym these days. We study the causal role of sleep in the emergence of mental illness in youth, with specific focus on psychotic disorders, but we have other interests that we're currently dabbling as well. In all the work that I do, I utilize sleep electrophysiology, brain imaging, as well as clinical and cognitive characterizations. My home base is in psychology, but I'm also closely affiliated with the Department of Psychiatry and the Neuroscience Institute here. So it is no surprise that there is a close link between sleep and psychiatric disorders. There are several epidemiological studies from many, many years ago that point out to comorbidity. And along with reviews that this co-occurrence is actually no coincidence. And even a recent study from your neck of the woods, for instance, shows relations between sleep duration and quality and a lifetime presence of psychiatric diagnosis. So there's definitely a link here. So here's my infomercial. The gist of what I'm interested in doing. We know that sleep disturbance precedes or even precipitates most major psychiatric disorders. We also know that sleep is critical for cognition, emotion, and brain development. So I argue that some, if not most of the cognitive and emotional impairment, cognitive deficits and emotional regulation impairments in patients are sleep dependent. And to even take it a step further, I believe abnormalities in sleep oscillations early in development may interfere with normal development and maturation of brain networks and make the person more vulnerable to develop psychopathologies later in life. But the limitation of our current conceptualization is that sleep disturbance is a vague and overgeneralized term that lacks a biological basis, and how it relates to the neurobiology of disease is rarely known or investigated, which is a missed opportunity. So the goal of my research is hopefully to identify precise abnormalities in sleep physiology and to be able to provide causal mechanistic explanations for the sleep-dependent aspects of the cognitive and emotional impairment and determine if sleep physiology can be a biomarker for detection or a treatment target, which is sleep is actionable. That's the good news. So for the purposes of this talk, I will focus entirely on schizophrenia. So I wanted to start with a very quick summary of the clinical picture here. Psychotic disorders are best recognized by florid psychotic symptoms, such as hallucinations and delusions. They're positive in the sense that they're the presence of some symptoms which are not typically observed. Negative symptoms are the absence or lessening of typical behaviors. Examples include lack of motivation, inability to feel fresh pleasure, or a paucity of emotional gestures. Lesser acknowledged, but nevertheless a core feature is cognitive impairment. And these deficits are typically evident before psychotic symptoms emerge. For instance, most patients report a significant drop in school or work performance long before other psychotic symptoms emerge. And our work in Boston with Dara and Bob show that some of this cognitive impairment is actually sleep dependent. So let's just unpack this. The research on sleep's role in cognition burgeoned almost 3 decades ago. The first was a study by Avi Carney in '94 on the relationship between REM sleep and procedural memory. Since then, the first cover feature was in 2001 in Science, which was the first of many. And eventually, the work on the cognitive role of sleep moved on to popular science outlets and even to Oprah magazine. So in a typical experiment at the behavioral level, what we observe is that memory performance across a wide variety of tasks is going to be better if learning is followed by a period of regular night of sleep compared to an equal amount of continuous wakefulness. And this sleep benefit, as we typically call it, is towards a facilitation of consolidation. Consolidation is the process that takes place between learning and retrieval, and it reflects the transfer of fresh and labile, unstable memory traces from the hippocampus into more permanent and stable representations in the cortex. Transfer process takes place offline when the person is not actively thinking of this new memory episode, and preferentially during sleep. And before I delve into the research on sleep physiology, although this audience is not new to this idea, let me just underline that sleep is not a unitary phenomenon. It's not you go to bed and a single chunk of behavior type of thing. Different patterns of EEG activity can differentiate between sleep stages, broadly separated into REM, rapid eye movement sleep, and non-rapid eye movement, non-REM sleep. These sleep stages are also associated with distinct brain states, such that neurotransmitter concentrations and direction of communication between brain structures changes across wakefulness, REM, and non-REM sleep. One line of evidence for the active role of sleep in cognition comes from human studies that shows relations between sleep physiology and offline changes in performance. For instance, for a word learning task, how much of the word learning a person retains over sleep correlates positively with how many slow oscillations or how many spindles a person has that night. Another line of evidence for the active role of sleep on memory comes from animal studies on memory replay. This is work out of Matt Wilson's lab at MIT, and I will keep this very simple and cartoon-like. If you record the activity from hippocampal place cells during a successful maze run in a mouse and identify the patterns of neuronal firing during learning and continue with these recordings during subsequent sleep, you will notice that the same neural assemblies are reactivated during sleep and in the same order. So even though the animal is sleeping, the hippocampus is rewinding and replaying the tape of the experience of the maze running. So these and other studies show that sleep is critical for cognition. Memory replay during sleep is thought to rely on three cardinal non-REM oscillations. These are cortical slow oscillations, thalamic and thalamocortical sleep spindles, and also hippocampal ripples. It's their precise temporal coordination that brings about memory transfer from the hippocampus to the cortex. So let me take you back to the story that I was telling with respect to schizophrenia. With schizophrenia, and particularly when patients are on stable doses of their antipsychotic medications, we do not observe any differences in gross features of sleep. It's very hard for me to convince psychiatrists about this, but this is just two tables from our many publications that compare sleep architecture and sleep duration and quality across patients and controls. And it's quite comparable. There's no significant differences. The difference we see is in the microstructure of non-REM sleep, particularly with respect to sleep spindles. These are 12 to 15 hertz waxing and waning oscillations that are characteristic of stage 2 non-REM sleep. Patients with schizophrenia have a specific reduction in spindles. Plotted here is the comparison of spindle density, which is the number of spindles per minute of stage 2 non-REM sleep between matched controls and schizophrenia patients, which shows a significant reduction. And it's not just the count of detected oscillations, but the spectral properties of non-REM sleep that is selectively disrupted in schizophrenia. So the black lines here are the power spectral density for stage 2 non-REM sleep in healthy controls, and the gray are schizophrenia. And perhaps it's easier to notice when we blow this up for the 12 to 15 hertz spindle range. Reduction in spindle density correlates with deficits in sleep-dependent memory consolidation. These initial studies of sleep in schizophrenia use the procedural memory task called the finger tapping motor sequence task, which is a very simple task. You see a sequence on the screen, and your goal is to type the sequence as fast and accurately as possible. Healthy controls show an improvement in this task from where they leave off their training at the end of the night and from where they pick up first thing in the morning. So that's the sleep benefit that we see typically in healthy controls. But on average, we see no change in performance, no improvement after sleep in schizophrenia. And although on average there's no improvement in the patients, their deficit in sleep-dependent memory consolidation correlates with their sleep spindle deficits. One of the first things I was curious with Dara and Bob was the task specificity or memory system specificity of these cognitive consolidation deficits. So I wanted to find out if memory in schizophrenia patients does not benefit from sleep across the board, or whether it's specific to tasks that rely on sleep spindles for their consolidation. So this was in chronic medicated patients. We ran a behavioral study over 2 sessions separated by 24 hours, and we investigated overnight changes in performance in tasks that tap on different memory systems. One of them is the word pair learning tasks, which rely on sleep spindles for their consolidation. So participants are exposed to word pairs. They study them, and this is followed by feedback training when they're given the first word and they're asked to guess what the second word is. If they're not correct, they're given the feedback, and this feedback training continues until they reach a criterion. Then we do an identical pre and post sleep test where they see the first word and they get no feedback for the answers that they give. In healthy young adults, as I said, spindle activity correlates with performance changes in this task. What we observed was patients with schizophrenia took longer to reach criterion. They had to have longer feedback training, but their pre-sleep test performance was comparable to the healthy control group. The only difference we noticed was with the difference overnight change in performance. With this type of task, the sleep benefit is protection of what the person has acquired pre-sleep. So in our healthy controls, they were not significantly different from zero in terms of overnight change in performance. So sleep was helping them retain what they had learned, but there was significant forgetting in the patient group emerging after sleep. This, for instance, I won't go into the details of it, in contrast to a perceptual procedural memory that does not rely on sleep spindles for its consolidation. Overnight changes in performance was quite comparable in the two groups. And if anything, although non-significant, patients were doing slightly better after sleep. The finding of reduced sleep spindle activity is quite well replicated across other labs in the US or the world. And importantly, it's not due to disease chronicity or antipsychotic medication, because it's found in antipsychotic naive early course psychotic patients who are later diagnosed with schizophrenia. So this is sleep monitoring. This is archival data from the University of Pittsburgh from a very long while ago. This is looking at their sleep polysomnography during their initial hospitalization with a psychotic episode before they were put on any kind of antipsychotic medication. So compared to controls, these early course patients had a reduction in sleep spindle density. But because we had access to medical records, we were able to see if in the long run they developed schizophrenia or another kind of psychotic disorder. And it turns out that it was only those who were later diagnosed with schizophrenia that had the spindle deficit at the baseline, at their first hospitalization with a psychotic outbreak. Again, this is part of the archival data, but young non-psychotic first-degree relatives of the patients also showed spindle deficits. So that points out to sleep spindles to be a biomarker of the disease, which has to have some genetic load, since it's not related to chronicity medications and is also evident in first-degree relatives. A couple of years ago, we wrapped up a clinical trial in which we targeted the sleep spindle deficit in schizophrenia and investigated whether we would be successful in pharmacologically increasing spindles, and if this increase would result in better sleep-dependent memory consolidation. So this was a placebo-controlled, double-blind crossover design, a labor of love. We administered 5 milligrams of a non-benzodiazepine hypnotic agent called azapiclone. It goes by the market name Lunesta, at least here in the US. And we did just a single 5 milligram dose of it right before bedtime to chronic schizophrenia patients and matched controls. This manipulation was very successful in significantly boosting spindle activity. We used a 58-channel, high density EEG array to measure sleep. And here, what I'm showing are topographical maps of the drug effect. At each electrode, for each person, we calculated the average difference in spindle density between placebo and drug conditions. And it's shown here as a percentage. Warm colors represent higher spindle density with the drug than with placebo, reflecting that there was an across-the-board increase in spindles, and more so in patients, perhaps because they had more room to grow. In all statistical analyses, we control for multiple comparisons, but this was such a strong effect that it yielded a giant cluster of the entire scalp, basically, for the drug effect. Participants also completed the motor sequence task before and after sleep for both placebo and drug conditions. And here I'm showing the difference in sleep-dependent improvement on the MSD task between drug and the placebo conditions. So there was no difference in our healthy controls, although we increased their spindles. MSD, overnight improvements on the MSD was very similar across the two conditions. And if anything, wait for it, consolidation was further decreased in our schizophrenia patients. And this was a rather surprising finding because after the drug night, the topography of sleep spindles was very comparable between the patient and control groups. So with just a single dose of the drug, we were very successful in boosting spindles, but we failed miserably at improving cognition, which is what we cared for. And to try to understand why that might have been the case, we turned to the orchestration among non-REM oscillations. As I said, it requires the precise temporal coordination of these three events for memory consolidation, memory replay to occur during non-REM sleep. Hippocampal ripples are not accessible, at least to us, with just scalp EEG. So we focused on the temporal relationships between slow oscillations and sleep spindles. To do that, we detected co-occurring sleep spindle and slow wave events, and we measured the phase angle of the slow wave at which the sleep spindle peaked. And we found, just like others, that the spindles favor the upstate of the slow wave. And this phase, this mean phase, did not differ between patients and controls. But what we found was that the drug in both groups was reducing the consistency of this timing. Again, this was a very strong, powerful effect. So in summary, we were able to enhance spindle activity, but focusing only on enhancing this one oscillation that's deficient did not prove to be an efficient strategy to improve cognition. I'm not working on this line of work anymore, but what they're doing is working in parallel with animal model researchers and looking at the triple locking of these oscillations and how candidate drugs might be affecting triple locking of these non-REM oscillations in animals before turning into humans for other drug trials. But a drug trial that's currently active in Dara's lab is for zolpidem ambien. This is shown to maintain the temporal relationship of these three oscillations, at least in mice. My interest is understanding the integrity of the neural circuitry involved in sleep spindles. As you all know, spindles are initiated by the thalamic reticular nucleus, and they're propagated to the cortex and synchronized through thalamocortical feedback loops. And the name reticular nucleus comes from its lattice-like structure. The TRN is actually a tiny, tiny sheet of neurons located in the thalamus-white matter interface, and it receives collaterals both from corticothalamic and thalamocortical projections. Its neurons are GABAergic, so its relationship with both of these projections is inhibitory in nature. Recent data from multiple sources converge on the TRN and the thalamocortical circuitry as culprits in schizophrenia risk. And let me just unpack what I mean by these data. You must remember this famous Manhattan plot from the 2014 paper of the Psychiatric Genomics Consortium. This was the first large-scale GWAS study of its kind in schizophrenia. And it identified a whopping 108 SNPs that are significantly associated with schizophrenia risk. One of those SNPs, the fourth significant one to be exact, was for the gene CACNA1I, which encodes a calcium channel that is expressed in the TRN. It's the thalamus and the TRN, is its favorite place to be expressed, to be specific. This is the spindle pacemaker, and its knockout in mice causes a selective sleep spindle deficit. TRN is implicated with another gene, PATCH-D1, which is associated with risk for autism spectrum disorders. You might know that autism spectrum and schizophrenia, being neurodevelopmental disorders, do have a remarkable genetic overlap in terms of their risk. This is work from Mike Halas's group at MIT. And PATCH-D1 is selectively expressed in the TRN of mice in early development, and it continues to be enriched in the structure throughout adult life. It's deletion results in spindle abnormality. So that's a reduction in the knockout group, which happens in the context of no change in total sleep duration in these mice. So both of these lines of genetic evidence suggest that some of the genetic risk for neurodevelopmental disorders implicates the TRN circuitry and might explain why patients have a deficit in the production of these oscillations. Another source of evidence that implicates thalamic circuitry in the etiology of schizophrenia is brain imaging. As I mentioned, the TRN is only a tiny sheet of neurons, and MRI, at least to our ability, does not have the type of resolution to be able to image it. We can image it in autopsied patients, in autopsied brains, but that takes several hours. And of course, that's only structural imaging. But instead, we're able to use functional connectivity MRI to delineate the thalamocortical network. Several data-driven or hypothesis-driven approaches have been used to characterize thalamocortical circuitry, but my focus today and in general has been on seed-based approaches, which look at the connectivity of the entire thalamus with every other rock cell, every other gray matter rock cell in the brain. So the finding, which was, for instance, proposed by Antichevich et al. Out of Yale, and this is a very large sample MRI study coming from consortium data. The finding is of abnormally increased connectivity between the thalamus and the primary sensory, primary motor, and primary auditory cortices, and reduced connectivity of the thalamus with the prefrontal cortex, so a pattern of hyper and hypo connectivity. This is a well-replicated finding, and it's also observed in clinical high risk and is in fact more prominent at baseline for those who later convert to full-blown psychotic disorders. So this is another consortium study from the same group, which compares healthy controls to those who were identified to be at clinical high risk for psychotic disorders and were later identified to be as non-converters or converters. And this is their MRI at baseline. Controls are the darkest color, as I said, who show a negative relationship, negative connectivity between the thalamus and the sensory motor cortices. And it's specifically the converters who were showing the hyperconnectivity pattern with the sensory motor cortices, both right and left side. So this connectivity pattern can be regarded as a biomarker. It's also been shown to correlate with positive symptom severity. In this particular study, we used 1 night of high-density EEG monitored sleep and a separate session of resting state functional connectivity MRI in patients with chronic schizophrenia and their demographically matched controls. So looking at thalamocortical connectivity, and mind you, this is a much smaller sample compared to the consortia imaging studies, I replicated the same pattern of hyperconnectivity of the thalamus with the primary sensory and auditory regions. With EEG, I also replicated previous findings of spindle deficits. Here's a topographical plot of group differences in spindle density with warmer colors reflecting more spindles and healthy controls. And we observed up to 50% reductions in spindles. That was widespread, but focusing primarily on frontal and central parietal regions. And this is the plot of group distributions for average spindle density and functional connectivity in between patients and controls. So what I did next was ask whether these two deficits are related. The blue here on the template brain are hyperconnectivity regions of thalamocortical hyperconnectivity regions. I ran whole brain regression analyses to determine which regions of the thalamocortical network show significant relations with spindle activity. This revealed a significant cluster, which is shown in red and yellow here. Particularly the green one is the overlap between the thalamic hyperconnectivity and spindle density correlation. So what we found in all of our patients happening across the spectrum is that greater sensory motor thalamocortical connectivity, the more hyperconnectivity a person has basically, is associated with reduced sleep spindle density. So in summary, what we found was we replicated previous findings of reduced spindle density and abnormal sensory motor thalamocortical hyperconnectivity. And we showed that these two deficits are correlated with each other. I had mentioned that spindles are generated by the TRN and are propagated to the cortex through thalamocortical connections. So a dysfunctioning TRN would result in a reduction in spindles, but let's think of the increased resting state connectivity between the thalamus and the sensory motor regions in patients. We hypothesized that the TRN is responsible for this abnormal hyperconnectivity because of its failure to inhibit the thalamus from sending unnecessary information to the cortex. That's our interpretation of how spindle deficits and thalamocortical hyperconnectivity are related to each other. But so far, I've only been talking about the sleep function of the TRN circuitry, which is propagation, initiation and propagation of spindles that plays a critical role in sleep-dependent memory consolidation. During wakefulness, the TRN circuitry acts as a gatekeeper of the flow of sensory information from the thalamus to the cortex, and it inhibits transmission of redundant sensory information to protect higher order cognitive function from interference, which can be considered pretty much like a searchlight guiding attention. And patients with schizophrenia anecdotally report difficulties with processing sensory information, and these deficits have traditionally been quantified with event-related potentials. So for a single or first brief auditory stimulus, individuals, healthy controls, show a prominent positive response. This is traditional ERP, so positive is down. They show a prominent positive response at around 50 milliseconds post-stimulus, which is termed as the P50 component. For a second identical stimulus that is temporally very close to the first, at around 500 milliseconds after, the response is attenuated. It's gated out as repetitive or redundant. And the most commonly used measure of sensory gating is the ratio of the magnitude of the amplitude of the second to the first response. So effective gating is reflected in a smaller ratio. In contrast to controls, patients with schizophrenia show the P50 for both the first and the second stimulus. And in their case, the gating ratio moves closer to 1. We had preliminary evidence for only a few participants looking at the relationship between sensory gating and sleep spindles. We measured, we characterized sleep spindles during a nap, and after they woke up, we asked participants to complete the sensory gating ERP experiment. This was young adult college students. And we found that better sensory gating, which corresponds to lower gating ratios, was associated with higher density of sleep spindles. And this preliminary evidence motivated our current work for investigating whether they have a shared neural basis. So this is my K award that we're working on wrapping up at the moment. And the idea was that, as you know, sleep studies are rate limiting for large-scale population-based or genetic study protocols because they're very expensive. They require highly trained technicians to collect the data and to analyze the data. They're also very time-consuming. So we wondered whether sensory gating could be utilized as a more scalable marker of TRN circuit dysfunction in schizophrenia than sleep spindles. We monitored sleep at the Clinical Research Center with high-density EEG. And the morning of the sleep recording, we obtained ERP measures of sensory gating. And on a separate session, participants completed MRI scans. Our participants were those with early course psychosis, defined as less than 3 years since diagnosis. So they were either patients with schizophrenia or other psychotic disorders, and their young first-degree relatives who don't have any psychotic symptoms, along with matched controls. So these samples were crucial in two aspects. Having the familial high-risk group allows me to identify what are trait-related alterations of thalamocortical circuitry, and having the early course patients with other psychotic disorders allows me to determine whether these abnormalities are specific to schizophrenia or can be generalizable to the experience of psychosis. So as I said, that's still work in progress, but with this study, we observed significantly reduced spindle density in the psychosis group compared to controls, and to my surprise, compared to familial high-risk group as well. When we separately examined the distribution of spindles in the patient group, we observed that the difference is driven by those who have schizophrenia and not psychosis of other reasons, which this replicates the archival single electrode study that I mentioned from many years ago. And that the spindle deficits are not generalized to the experience of psychosis. They seem to be specific to the etiology of schizophrenia. We observed a very similar pattern for sensory gating compared to controls, and again, compared to the first-degree relatives. Psychosis patients had abnormal P50s, indicating reduced sensory gating. But this time for this, we did not find any differences between the schizophrenia and non-schizophrenia patient group. We also observed a modest but significant correlation between sensory gating and sleep spindle density. So in full disclosure, am I confident that P50 ERP experiments can be used as a tractable readout of TRN function instead of sleep spindles? Not quite, but at any rate, both of these indicate show moderate but significant correlations with psychotic-like experiences in our entire sample. What I'm plotting here, these are like last night thrown together slides, but this is using Chapman scales. In other words, the Wisconsin schizotypy scales that measures magical ideation, and this is for the entire sample. And those with the highest endorsements of these psychotic-like experiences, in turn, are the ones that have lower sleep spindle density and worse sensory gating. I'm also looking into functional connectivity of the thalamocortical circuitry in these participants. And what I observe is hyperconnectivity of the thalamus with the primary auditory cortex in both familial high risk and also psychosis groups. And the future goal for this, what I'm working on right now, is to identify thalamocortical connectivity correlates of both P50 sensory gating and sleep spindle densities. But I wanted to end this talk so that we have quite a bit of time for chatting. But I also wanted to end this presentation by talking about what we're up to here in Iowa. I'm quite committed to understanding the role of sleep in terms of comparing risk for psychotic disorders or for acting as a resilience factor. To that end, I teamed up with my colleague, Amanda McCleary, in my department who's also a schizophrenia researcher. And as a major plus, she's from Toronto. She worked with Gene Addington before coming to the US. The two of us recently, as in a few months ago, launched a clinical research program. And our goal is to provide evidence-based psychosis risk assessment to youth who may be experiencing unusual changes in their behavior, thoughts, or perceptions. And if the individual meets criteria for a psychosis risk syndrome, or if they're in the early phases of a psychotic illness, we offer longitudinal monitoring of their symptoms every 3 to 6 months. And our aim here is to be able to catch the onset of psychosis or functional decline as early as possible and expedite the person's and the family's access to appropriate psychiatric treatment and support. And as our program grows, hopefully with funding, we eventually want to start offering interventions through research studies, for instance, CBT for a psychosis, or sleep hygiene training, or CBTI for insomnia, along with patient and family support groups. So these participants are quite valuable for research, and they will be invited to participate in our research studies. And the next step that I'm tackling in this population is to be able to map out the developmental trajectory of the thalamocortical sleep spindle circuitry in at-risk youth. Our assessments include genetic assays, MRI imaging, and the use of wearables to obtain as much data points as possible on sleep macro features and sleep micro features. We use wrist-worn actigraphy watches. So this is a picture of the Philips Active Watch. I don't know if you guys are following the news, but they discontinued it. So I'm quite bitter about this whole actigraphy business. But we use both wrist-worn actigraphy watches for objective evaluation of sleep quality and duration, and also a headband-style EEG monitors to be able to characterize sleep spindles and other non-REM oscillations in these participants for as many intervals as possible. And I'm very much interested in understanding the role of sleep oscillations in the developmental trajectory of thalamocortical circuitry throughout adolescence, from before to after puberty, where we know there's quite a bit of changes happening at this age range. I'm also committed to understanding sleep-related biomarkers for risk as well as resilience in at-risk youth who may or may not convert to a full-blown psychotic disorder. So data coming from those who do not convert is as valuable as the data from those who do convert. So in closing, I talked about my research on schizophrenia, but my lab in Iowa is not limited to just this pathology. We're interested in other psychopathologies, for instance, pediatric anxiety, and more recently, stroke and the role of slow oscillations in stroke rehab. And our approach can be applied to other disorders. The pipeline for us is to start with a disorder where sleep disturbance and cognitive or emotional regulation deficits have been reported. Next, we aim to identify exactly what these sleep disturbances are. Are there abnormalities in sleep oscillations? Is that an issue with sleep architecture? Could it be a circadian shift or insomnia? The next step is to determine the relations of the sleep abnormality to cognition and emotional processing. In other words, asking what aspects of the cognitive deficit in this particular disorder is sleep dependent. Then I like to examine the neural circuitry involved in this particular deficit. For instance, the brain structures that are generators of a particular oscillation, or brainstem nuclei, for instance, if it's a problem with sleep-wake generation or stability. And then we also turn into ERP sometimes, as I mentioned, with the sleep spindles to tap on the wake functions of this circuitry. with the ultimate goal of providing a mechanistic explanations of the role of sleep in the pathophysiology of neuropsychiatric disorders, and also to possibly devise clinical trials that will precisely target the sleep disturbance. And as I mentioned, I make use of several tools that include clinical and diagnostic interviews, cognitive experiments, sleep EEGs, MRIs, and ERPs. In closing, I would like to acknowledge my lab, who have been working round the clock very hard in trying to collect these boutique type of data. It's quite exhausting to be able to get all of these, and also acknowledge my mentors, Bob and Dara in Boston, along with support I've received from the NIH and internal funding here at the University of Iowa. So thank you very much.
Thank you, Dr. Baran, for that lovely, amazing talk. It was very fascinating to hear about the effects of the primary drug administration on the spindles. I know it's different for cognition, but I also think the program that you are offering for the young adolescents to learn about their early diagnosis is also amazing. I welcome everyone right now to ask any questions to Dr. Baran for the next few minutes we have. Kindly use the raise hand function, and when called upon, please unmute and ask your question. Sean?
So great talk, really beautiful work, and congratulations on all the progress so far. I think it's really exciting. As you know, I have a deep and abiding interest in TRN as well, and I think one interesting follow-up would be to look at some of the implications of the recent TRN, the thalamoreticular model that we recently put out, and the differential depolarization between TRN versus the thalamus, and some of the effects on spindle generation there. And it'd just be, I think, worth a follow-up conversation, just to look at the interpretation there. But the core question that I had is, of course, one interest, and I'm sure you share that, is what's a really good scalable model to measure this, broadly and ideally during wakefulness. And of course, you've got the ERPs to do that. But I'm wondering, what do you think the chances are of kind of a EEG signal, not necessarily with ERPs, and looking at other rhythms that are generated by TRN, alpha range, and has there been already a clear association in those individuals showing reduced spindles during sleep, that there's a corresponding alteration in any frequencies during wakefulness in the EEG?
That's a great question. Another possibility we're looking into is gamma, particularly as it relates to auditory steady state responses, which might be tapping on this excitation inhibition differences. We're working on that. We got the data, we're working on analyzing it. So I don't know, but as I was reading your model, the first thing that came to my mind was maybe they can answer this all, what's happening with the TRN during wakefulness question.
Yeah, it'd be great to discuss further. Thank you.
Yeah.
Anyone else? Taha?
Hello, Dr. Baran. Thank you so much for the great talk. Very enjoyable and very, very clear on especially the spindles and their roles in memory and how that could be changed in schizophrenia. Now, the question that I would have is there a change in the spindles that we could see among the progression of disease, across the progression of disease, from early stage to late stage? Does the do the parameters of the spindles change, and how that would correspond to changes in the TRN and with respect to the phenotypes? Thank you.
That's a great question. So there's no longitudinal study of what's happening to sleep oscillations throughout the course of the disease, right? All I can say is with these cross-sectional data, we see deficits both in early course and also later. But work out of Matt Walker's and Bryce Mander's labs are showing that aging is also associated with quite a bit of decline in sleep spindles and slow oscillations, right? And an interest, something that I'm hoping to submit as an art pretty soon is looking at age-related changes in NREM oscillations in schizophrenia to see if there's some sort of an accelerated aging or accelerated path to dementia that these patients are exhibiting.
I see, I see. So that would be in relation with N3, basically low frequency stuff, or would it be?
Exactly. So not just the thalamocortical spindle oscillations, but also slow oscillations and their relations with their timing relations.
Yes. Okay. Thank you so much. I appreciate
Yep.
it.
Yep. Thank you for this question. Come review my art when I submit it.
I would love to read it. I'm a grad student here, and my work actually focuses on sleep as well, sleep EEG. So very interested in your work always. Thank you.
Awesome.
Thank you. Thank you, Daha. Anybody else? Any other questions? Mohammed?
Yeah, thank you very much for this interesting talk. So I'm not really familiar with sleep spindles as I haven't worked in EEG for a really long time. But from what I understand, it seems that even after restoring the sleep spindles using like medication, it did not really affect the memory consolidation in schizophrenia. So it seems that like this is more of like a marker or a symptom rather than something that actually mediates the memory consolidation. So the question is like, what is the precursor that would be that maybe we are looking like maybe the medication is targeting like the after, like not the reason of the memory consolidation lack, but the like the symptom itself, so sort of like alleviating the symptom of the sleep spindles. So I was wondering, like, what are your opinions about what is actually happening in the beginning that the medication is not affecting that could?
I see. So that's one way of interpreting this. Our thinking was that everything happens in an equilibrium, in a homeostasis in sleep, right? So just pharmacologically increasing one type of oscillation, you have to be in close control of what's happening to other oscillations. To what extent are you increasing these oscillations, right? So our conclusion was that it's not that spindles were not a precursor per se, but when we increase spindles, we messed up with their temporal relationship with other oscillations. If anything, we messed up with the ideal milieu that memory consolidation could occur. Otherwise, spindles known to have a causal role in memory consolidation. Spindles are also thought to be, based on recent studies in epilepsy patients, for instance, spindles are thought to be the leading oscillation for all of these three non-REM oscillations to align.
Okay, that makes it a lot clearer. Yeah. Thank you.
Thanks, Mohammed. Thank you, Dr. Baran. Anyone else? The next question? Okay. Thank you so much for your amazing talk. We would love to have you again and discuss, more things as they come about. So I think that concludes our talk today. If anybody wants to have any questions, please email them to me, and I'll forward it to Dr. Brown. Thank you.
Yep, I'm also very Googleable. Yeah, thank you so much for the invitation. I really enjoyed this interaction.
Thank you so much. Take care.
Bye-bye. Thank you, Rabia, for your help. Bye-bye.
Thank you. No problem.